¿Ya estás grabando, Fer? Oh. Well, fair enough. Here we are. Here's our team. With Carla Gomez, Carlo Ramón Hinojosa, Edgar Fernando Sánchez Briviesca, and me, Hector Delgado. And today we are presenting our journal club number 2. Today we are exposing more specific papers focused towards our final project, which we have previously discussed, will be centered on the use of biosurfactants to treat/prevent reduce intensity of COVID-19. And this time we will begin our presentation with Edgar Fernando. Please begin.
Hello, I'm going to present you a very interesting article it's basically justifying our project. It's an article that explains how surfactant-based prophylaxis and also therapy could be a mean to stop the COVID-19 pandemic. And they say that it's a possibility, and they further explain how it's going to work. This was just published this month in Med Hypothesis, so we had no way to know this. It was actually published in July, but it had no article. It was just online, and finally got its publication in a journal on this month of this year. So let's begin. The authors start explaining about COVID-19 and how it has affected the lifestyle of basically the entire world, and how many lives have been taken and all that we all know. But they don't make a really good job of explaining how the surfactant could work against COVID. So I did a little bit more research, and I found this. I'll just explain very quickly. Well, we know the virus consists of a lipid membrane that has proteins on it, and it encloses the vital proteins and, well, the positive sense RNA that it's the source of infection. Then the amphiphilic nature of biosurfactants will allow them to interact with this hydrophobic domain of the capsid of the virus and then degrade it and disabling its infectivity. And the advantages of biosurfactant is that compared to their synthetic counterpart, they are obviously, well, degradable. First of all, they have greater emulsification activities, work in a broader range of temperatures, and they have low toxicity compared to the synthetic part. And then we have another thing that they don't mention in the article. It's also the critical micelle concentration. That is basically when you have biosurfactants dissolved in a liquid, they start to form and begin to concentrate until they reach a mycelic form. So they gather up, and the hydrophilic part is going to be inside, and the hydrophobic part is going to be outside, and they form these little spheres of micelle. So no matter what happens, if you add more and more surfactant, they're not going to have a bigger effectivity in decreasing the surface tension. Okay, the article starts postulating this, that they say that a simple biosurfactant-based gargle will be enough to stop the spread of the pandemic. In addition of this gargle, the surfactant-loaded throat point, mouthwash, nasal drops, and eye drops could be also used for prophylaxis, also using biosurfactant, and this will be particularly useful in high-risk categories and health workers. In addition to prophylaxis, they state that surfactant therapy would be a beneficial treatment in COVID-19, and basically they're stating that with biosurfactant that you could stop the pandemic. So how do they tell you that it's going to work? They hypothesize that prevention with gargles would work like this. Basically, is the viral entries to the eyes, the nose, and the mouth, and the choke point in all of those entry points is the gargle. So you're just gonna make a mouthwash, and gargle it, and it's gonna stop the virus from spreading. Since you're gonna have it in suspension or adhere in the epithelium, so it's gonna protect it from the virus. They state that the surfactant is gonna degrade some of the surface protein, but maybe it's not enough concentration in order to degrade the whole membrane. So they just inactivating its surface proteins that are the cause of infection, or the first way to infect the cells. Then they state that surfactants have been used in lung and tracheal infections and have not caused damage to the cilia in lungs and trachea. And so they say that you could dose a patient with surfactant, get it to the alveoli, and then protecting them from inside the alveoli. And then they also say that if the surfactant it's capable of, it could go through the epithelium and get into the interstitial fluid, and then go to the blood vessels. So they're stating that a surfactant is going to be capable enough to go through this tissue and also protect you from the vessels. And that's how it's going to work. Basically, it's going to go through your lungs and then go through your blood vessels, the surfactant, and inactivate any particle that is going to, any viral particle that will infect you. And they, as I stated, they suspect that it's going to prevent the attachment of the virus through its spike glycoproteins. So they are not stating that it's going to break down the virus as if the whole virus. Just interfering with the spike glycoproteins, resulting in their destruction eventually. And well, then they state that surfactant therapy is already used in respiratory problems. Surfactants, as we know, in humans have innate host defense during infections. There has been artificial surfactant that had worked against H1N1 virus in mice. So we already have an in vivo test for surfactants. It's artificial. It's no biosurfactant, but it's something. Proven the lipid part of some surfactants have been capable of inhibiting the H1N1 influenza virus in vitro, and recently they have, and it's a photo on the right. They have there has been a study that is proposing to use bear bile salts, that is a biosurfactant, a chemical biosurfactant, not as well. It's a salt. It's not as a rhamnolipid or another type of surfactant, but still, they're stating that using bear bile salts is going to be successful against COVID-19, and with also all these cases in its favor, they state that they could be a promising therapy against COVID-19, and steps of replication, yes, whether it is inside or outside the cell. That's what they're stating with this information. And then they provide us with a list with potential surfactants using pharmaceutical and that have been already approved in industrial and clinical use. Some of them are approved by the US FDA, and some of them have been approved like an extra ingredient, not as a treatment like so. And this is basically what they have found in different uses. And there's all the reference that are probably going to be really useful for a project. And now we're stating about the prophylaxis that is protecting the host from the contagion of the virus. So what about the virus spread if you already have it? Basically, the biggest point of infection in COVID-19 is the droplet form. That is basically a little droplet of saliva that is going to spread through the air, but it doesn't have a really big reach. And so what they're stating is that you could prevent contagiousness from patients by doing gargles. So if you have a patient that has COVID-19 already, you give them gargles, and their saliva is going to be protected from infecting another patient. And then this also could be in the form of the eye droplets and all the methods that is stated in the beginning. And then if there's virus spread through blood and other bodily fluids, as they state, if you give surfactant to the lungs, it's going to go into circulation in the body, and it's going to destroy the virus, and it's going to be non-effective. And they close up saying that they believe that the surfactant-based methods could prevent the spread of the pandemic. Moreover, if they demonstrate the utility of this method, it will be a good solution to stop viral infections in the future. So it's not going to be only focused on the COVID-19 pandemic. And then they state, how should you do the testing of their hypothesis? So first, you need to identify the surfactant and its concentration or dose for its use. Then the critical micelle concentration will be the base point to start for every biosurfactant. So as I told you, is the concentration at the point that even if you add more surfactant, it's not gonna increase its effectivity. And then the second one is to check its safety for the intended use. If it's a gargle, if it's for the eyes, if it's for the lungs, so you need to check every single type of toxicity. And with in vitro studies should be enough to do a primary screening of all the stabilized surfactants required, and then you should do a really detailed study to check the effect on the spike protein and how it's degrading it and how it's going to work in the cell. And they say that you should divide the focus of the investigation into types: the prophylactic and the therapeutic effect. So one is more complicated than the other. Obviously, therapeutic could be a little bit more complicated to study. And that's it. Questions?
I have some comments and kind of questions.
Yes.
The first comment is, even though it's stated like a salt, the bile salts, they are only the saponification of some lipids. So indeed, they have the same nature of lipidic amphipathic compounds. So any of other soaps have the same salt composition. That was just a clearing. And the other one is, if the viral replication is most, it's higher in the upper respiratory tract, why do they stated that gargles are the most effective mechanism to stop viral spreading instead of a direct application into the nose? Because they are deprotecting one of the main zones of viral replication, that it's the nose, the pharynx, and with a gargle you cannot reach the larynx.
They don't state it clearly, but my hypothesis is that it's a choke point for every connection of the respiratory system. So even though if you start to replicate the virus in that part, maybe if you have the gargles that are in the choke point of all those respiratory connections, it's going to stop them from going to the lungs and make the acute respiratory syndrome.
Okay. But then you're protecting yourself, but not inhibiting to spread it.
No,
right?
No. Yep. Yes and no. They state that if you are a patient that already has it, if you do mouth washes combined with gargles, your saliva is going to be inactivated. They don't state about the the nose, the nose cavity, but they do say it about the mouth and the throat.
I have another question about the blood, the biosurfactant going into the blood. Did they mention any trials or any studies which support this hypothesis, or is it this de novo hypothesis which they want to eventually test?
If I'm not wrong, they had some articles stated to support that information. I don't remember which. I actually didn't check each one of the articles, but they say that it has been proven that it could work, at least in vitro or in vivo, but I don't know if in humans.
Okay, and to the list of biosurfactants that is proposed, do they mention the critical micellar point or not? Just quickly, do they or do they not?
No, they just state that the articles that the information came from, but I suspect that it's not gonna be. That's the critical micellar concentration depends on the surface and then the media that you're putting it. So it's gonna change for every tissue or every single part that you're gonna study. So it's not completely interchangeable between different tissues.
Okay. Thank you.
I have another question, kind of the same of Hector. You mentioned at the first part of the presentation that there was like no toxicity in lungs and trichal cells. Did they mention like some protocols that they do in order to obtain that result?
Yes, they just stated it. The actually this review was just, well, this hypothesis is not a review. It's really just like really short. They didn't explain it deeply how the mechanism worked. They just state that they found it and it could work. It's not like a really robust explanation of every single mechanism that they stated in this article.
Okay.
But yeah, probably yeah. They state that it didn't damage either of the cells. So there's the article is cited there.
Okay. It's so.
Well, fair enough. If there's no more questions, I believe we can move on to the next presentation, and I do believe that's gonna be me. If I could share my screen. I don't know if you can see it now.
Yes.
Fair enough. Just one quick question, Fernando. Is the session recording? Because I do not see it.
Yeah, I'm watching it. I'm watching it record.
Yes, it is.
Okay, because I couldn't see it, so I had doubts about it. Well, here we go. I decided to do more research into how the virus and its effect is quantified in vitro for further studies in laboratory as well as tissue culture, because in order to develop any sort of protocol or any sort of trials to determine the efficiency of any possible treatment, I won't say specifically the word drug, but any treatment, we need to be able to have enough virus to properly study them in the correct types of cells and to have trials so we can see the effectiveness and the effects of this virus on the cells. And the paper I found, which is accordingly called Growth Detection, Quantification, and Inactivation of SARS-CoV-2, goes perfectly into detail about this topic. The first part of it is the propagation of the SARS-CoV-2 in vitro. And well, in order to begin this process, we need to start by a stock obtained through human sampling. We need to have an initial stock from which we can culture and propagate our virus. The authors mentioned obtaining a human sample as the initial stock, and they made a review upon which cells can possibly be used to study this virus, and they found that African green monkey cell lines are some of the best cells which allow for the propagation of this virus. And rightfully, I do believe that for our project, we will focus on our study on this type of cells, just for the part of propagation and maybe some initial trials. But the cell line that we would be using would be African green monkey cell lines, Vero E6, which has been studied that it is able to receive and to propagate and is susceptible to SARS-CoV-2. Here we can see on the right side a propagation trial of the Vero E6 cells compared to the SARS-CoV-2 cells. The difference in the coloration arises from the cells rupturing, and those white spots that we can see are exactly the areas where the virus has actually bursted the cells, and therefore that is from where we can obtain our initial stock for further propagation, inoculation of tissue cultures. Afterwards, we need to be able to detect and to quantify our amount of infection. I read in other papers which I will just mention quickly that there has been a study and the amount of virus, the viral load has been correlated to higher mortality. Therefore, that's one quick way we can see that if we are able to reduce or decrease the amount of viral load, this could be a potential treatment, not a cure, but a potential treatment. The rate of infection won't be as high. But returning on the topic, we have to be able to determine that we, in fact, have our coronavirus in our cell cultures. We have to be able to quantify it. And the author goes into various methods that they do for this. First, for the initial detection of the cell infection, we can use real-time PCR assay for SARS-CoV-2, extraction kits and PCR kits for this type of detection, like real-time PCR, are widely available for clinical trials, and many pharmaceutical companies already sell kits and primers for all of this. And then, in order to quantify the severity of our infection, we saw two possible ways to quantify it. The initial one is quantification through plaque assay or through focus forming assay. The first one, which is the center image you can see with the most color in blue, is a plaque forming assay. And what it does is quantify the amount of white dots that are being formed, which analytically count and thereby calculate how much of a viral infection is occurring by seeing how many of the cells are being bursted by the coronavirus. We start with an initial culture that is close to confluence, and after infecting it, we can study how that infection occurs through our Petri dish. And one benefit of this technique is that it's really quick and it's relatively low throughput in order to study the effects of it. The second technique is the focus forming assay, which begins with the same principle, but then requires for it to be analyzed through antigen, with antigen fluorescence and antigen detection, sorry. We do the same thing as a plaque assay, but afterwards we transfer our sample into a nitrocellulose membrane and do an immunological antigen assay in order to determine where and how much of a protein of COVID is detected. And both of these methods can be done complementarily. They can complement each other, but they both quantify the same thing. And the authors go further into detail as to being able to detect SARS-CoV-2 antigen by flow cytometry, but I won't go into further deepness into this because in my close to professional opinion, I would do believe that the first two assays can be correctly done. And also to determine that we have concluded our test and we don't want a further spread of this virus, the authors go into outgrowth assays for validation of inactivation methods and list the different compounds that can be used in order to arrest the the coronavirus to arrest and inactivate the coronavirus within our samples so that we can quantify them without there being any changes from these samples. Further cell lines can be studied, but I do believe that all of these can be useful for our project as a general protocols and techniques for the project and the different trials we can do. Here is my bibliography, the paper, authors, as well as the publication date. And I do believe that this is it. Any questions?
Yes, in the part of the immunoassay that you mentioned.
Yes.
Are there any antigen? Are there any
monoclonal antigens for the detection of the coronavirus?
Primary
antibodies?
Antibodies. Thank you.
Yes, I do believe that there are. I do not remember exactly which antibody they use, but it is more mentioned as a technique that is widely available to do these kind of protocols. But they do not go into depth into which antibody they use. They do go into depth into the media and incubation times. The general protocol is properly established, but they don't go into any further deepness about the antibodies that are being used.
But they are in the market.
Yes, they do exist in the market, and they are actually available. Okay. But yes, I do.
And the other question is, yes. Do they mention what type of RT-PCR they made?
They do go into detail. They even explain the primers that they utilize. Okay. I did not go into further detail into this because, well, I think that you all know how PCR works, and there's available information about this in many areas, and they just mention it as a technique for proper detection and to determine the proper inoculation of our samples. They do mention the primers, from where do they get them.
They don't mention if they use like a quencher and a fluorescence probe, or if they use a fluorescence dye.
I think they mentioned the kit they bought. I think they acquired it from Biorad or Pfizer. I do not exactly remember, but it's a commercially available kit.
Okay.
Fernando, Carla, any questions from your behalf?
Yes. Do they state if the detection could work in other type of cells apart from their E6?
Yes, they do mention a list, and they do mention various cell lines that can be used. Further studies that I read mention more cells. Human epithelial cells can actually be used to study the virus, but I just mentioned this because these are some of the ones that the authors explained and used for their trials and were successful in both propagation, detection, and quantification. But there are more lines than these ones. Carla, any questions?
No, I don't have any questions.
Any other ones from both of you, Chuck and Fernando? Well, in that case, I stopped sharing my screen, and I think it's either Carla or Carlos' turn.
I think it's my turn. No?
Yep.
Where can I put? Do you know where can I share my screen?
It's just right besides the microphone. It's the little square with a straight up arrow.
Thank you. Are you watching already? No.
Maybe not yet.
No?
No. Okay, we can see it now.
Okay. Well, I'm going to talk about the in vitro surfactant structure, toxicity, and the relationship in the application for surfactant use in sexually transmitted infections for prophylaxis and contraception. Well, a few introduction is that all these emerge from the need to develop new topical treatments for prophylaxis against sexual transmitted infections that at the same time it could be safe, cheap, effective, and practical to use and to apply for women. And they have been researching the development of formulations containing specific antiretroviral drugs, but however, as we know, some viruses are highly mutable. So the main objective was to demonstrate the in vitro effects of surfactants concentration, as well as their how it's related with the exposure time and structure of the viability of mammalian cell. The type cell line that they used was mammalian cells, mainly fully polarized and confluent epithelial cells, and also confluent but non-polarized epithelial. The surfactants they used was Triton X 100, monolauril, DDPS, SDS, and the other three that they are mentioned there. The methodology that they used to prove the toxicity towards these Lyme cells was by MTT assay, 24 hours post-exposure to surfactants. And this methodology or this test is based on the cleavage of the yellow tetrasodium salt MTT to form a violet purple formazan. So a decrease in the number of living cells results in a decrease in the metabolic activity in this test culture. So this decrease is directly correlated with the amount of violet formazan, and it can be monitored by the measure of the absorbance. So practically in this protocol or in this test, we will see a result determined by a colorimetrically. There are some remarkable facts that, as they mentioned, the critical mycelium concentration, that is the minimum concentration of surfactant from which micelles spontaneously form in a dissolution, and it could involve a cell membrane destabilization or a destruction. They also mentioned that the toxicity depends also on the chemical nature of the polar head of the group of the surfactant. And mainly the cationic surfactants may work as a bactericide at concentrations that are not harmful to polarized mammalian epithelial cells, but to the virus. So in the other slide, we can see the results. They test the toxicity of 4 different commercially surfactants with different concentrations. They use 10%, 50%, and 90% in different periods of time. That it was their exposure time. It was 20, 60 minutes, 180 minutes, and 545 minutes. So we can see that for TX100, DDPS, and SDS, the toxicity was not observed up to concentrations close to the critical mycelium. Also that the toxicity of CD-TAB was at concentrations that were much lower than its critical mycelium. And despite their low toxicity, those 3 surfactants, the DDPS, SDS, and TX100, were shown to have neither bactericidal nor spermicidal activity at concentrations that were not harmful to epithelial cell. And the objective that they do this to prove this toxicity in these cell lines, that was MDCK and CACO2, the cell lines, was that they observed that the vaginal columnar epithelial cells is the primary site of damage in the use of surfactants. Then they use the same methodology, but with different cell lines. That was HeLa cells. And they choose these cells also because it's a human cervical cell line. And they saw that here, TX100, DDPS, and SDS induced cell toxicity at concentration close to the critical mycelium. Monolauril did not induce toxicity at concentrations up to 10 times its mycelium, critical mycelium. And they came to the conclusion, and they observed that in HeLa, the toxicity was 10 times less than in the MCDK and CACO2 cells. Yes. So the results of all these surfactants was that they reveal concentrations and time-dependent toxicity effects, but exhibit different degrees of toxicity. And this, as I said before, it depends also on the chemical nature of the polar head of the surfactant. The Triton X100, DPS, and SDSC toxicity was not observed up to concentrations close to the critical mycelium concentration in both in non-polarized epithelial cells, HeLa, and polarized epithelial MDCK and CACO2 cells. On the other hand, that the cationic surfactants are probably toxic at more suitable levels, toxicity being at concentrations that are not sufficient to cause significant damage to physical integrity of the membranes of the cell, but they are the most harmful to the viruses. So the cationic surfactants are the best option in the usage of prophylaxis and prophylaxis to sexual transmitted infections. So that's all. Do you have any doubts?
Yeah, I have one question.
Yes.
How accurate do you think that the results of this study are, as they use a cancer cell line instead of normal epithelial lines of the vagina? Because some of them can be really tough to a lot of conditions. So what do you think? Is there an accurate result?
In what cells? In HeLa?
They use HeLa and CACO. But those are cancer cell lines. Why didn't they use epithelial vaginal cell lines?
No, yes, they use also columnar epithelial, the CACO2 and the MDCK. They are also epithelial columnar cells.
But CACO and HeLa are cancer cells.
Because I think it is related to the... No, I don't know why. No, I don't know why did they choose the cervical lines.
Okay. I think that they chose them because their accessibility, they are cheap and they are hardly replicated, but they might have a bias as they are cancer cells instead of normal tissue cells.
Okay, thank you for the comment.
But I don't really know.
Okay.
Probably I could investigate it, and the next time I'll tell you why they choose that or how it could be valid those results.
Okay. I have another question. Since we are working with biosurfactants, do they mention anything about working instead of synthetic surfactants, that is Triton X, Monolauril, and DPS, SDS? Do they mention anything about working with biosurfactants? since they, as I stated before, they have lower toxicity and they have well, they're better in a lot of ways.
No, they didn't mention, but I don't think if they have proved that biosurfactants could be not fully used, but they give the results that against sexual transmitted infections. I don't know if there are results that they compare biosurfactants or synthetic surfactants, but they didn't mention.
I have another question. Do they propose any sort of application or how this could be used for, or if they are directly applied to the possible patients? Do they propose any sort of trials in humans with these types of surfactants for any type of treatment of any disease?
In this particular, yes, because it is like mainly for sexual transmitted infection for women. So the methodology is practically with your hands to apply it. But in this case, it is special because it's mainly for that. I don't know how to say it. It's not like very important to have a protocol like with other biosurfactants, because in this is only topical, and they mention that they are like searching new topical formulations that they were easy to apply and to store.
Okay, thank you.
Yes.
I have a last question.
Yes.
You mentioned that the most cytotoxic surfactants they figure out were those with cationic nature, right?
Yes.
Which of those four, I think that they are four, surfactants have that kind of nature, and which other natures have the other ones, for if all were cationic, or I don't know.
Oh, I write it down, but I think, let me search it because I have it. Give me a minute. The SDS is anionic. I think the monolarine is anionic. Yes, monolarine is cationic. Cationic is also the SCN tab. And triton. And triton X100 is non-ionic.
Okay.
Well, in that case, then we can continue with our last presentation. Carlo, as soon as you can, well, please begin.
You can see my screen?
Yes, we can.
Yes.
Okay, let me project it. Let's see. You see it in a full screen?
Yep.
Yes.
Okay. The article I want to present to you is Animal Models for COVID-19. This is an article review published in Nature. These are the authors, and this is the link. First, they have a really brief introduction, and I think that this is really important because in order to know which type of animal model you have to choose, you need to know very well the nature of the virus and the nature of the disease itself. Also, I want to state clearly that this review is made possible by the WHO development. They have a team that it's called the COVID-19 modeling team of the WHO, that it's only focusing in animal models to study this type of disease. So let's review some of the most important aspects to study COVID-19, and is that, well, we know that this disease is stratified in age. Over 65 years, the mortality increases with 1% each year up to this age. The range of mild symptoms include fever, cough, and dyspnea, that is the difficult of breathing, and myalgia, that it's ache. Also, they have its main replication zone. It's the upper respiratory tract, as I mentioned in Fer's presentation. The disease also, it's progressive and cause severe damage such as pneumonia, acute respiratory distress syndrome, inflammation, alveolar damage, edema, and problems in the membranes. Also, it's not only a respiratory disease, as in a whole body, there can be a lot other type of issues, such as coagulopathy, elevated fibrogen in blood, D-dimer also, and also an increased thrombus generation that can cause a lot of harm. So these are the aspects that we have to review in order to choose a correct animal model. So the first of all the models that we can think about, it's the use of mice, as it's the most widespread animal model. But mice doesn't have a natural ACE2. They have their own ACE2 protein, but it's not compatible with the SARS-CoV. So we have to make some changes in order to study the virus in mice. First option is to use virus adaptation mouse. And this strategy is to put the virus in mice that is already diseased, that has the disease of other RNA virus, and expect them to combine, so to create a new virus, a SARS-CoV with a new RNA virus. So their mixture has the ability to infect the mice. This, first of all, is really dangerous because you're creating a new virus. And also, it's seen that their combinations are really mild disease and can bring wrong conclusions, for example, in the antibody studies, because SARS-CoV is no longer SARS-CoV. It's mutated with other type of RNA of a foreign virus. The other strategy is to express the human ACE2 in genetically modified mice. Nowadays, we have 3 type of GMO mice. One is this line, that it's with a promoter for called K12 of epithelial cells. Other with a universal promoter, that it's from a cytomegalovirus with a chicken actin, and well, they mentioned that it's universal. And an endogenous promoter of the ACE2 of the mice with the ACE2 human protein. And the other strategy is to humanize the mice, that it's putting some cells of the human into the mice, and let them grow inside the mice. But this strategy, I will explain it later. With the transgenic ones, the range of damage in the mice, it's really wide. They can go to mild symptoms up to little symptoms, including encephalitis, but they don't show neurological infections. And also, when you express the ACE2 protein of the human in mice, it is expressed ectopically, so you can see some changes in tissue and cellular tropisms, and those changes can bring some wrong conclusions that you're expecting that some tissue is having a damage, and it is not. It's only expressing a foreign protein, which give another type of nature in its morphology. And the last approach, as I mentioned, is to humanize mice, and one of the example is to induce amyloplasty, that is the whole killing of its bone marrows of the mice, and put some human bone marrow into the mice. So as they don't have hematopoietic cells, they cannot have an immune response to the new human cells, and they start replicating. But this protocol is only functional in the study of SARS-CoV in immune cells. It's not functional for other type of cells and tissues that you might want to study. The other strategy is to use hamsters. And hamsters are really good models in the study of SARS-CoV. They show a lot of symptoms up to mild to moderate. They have weight loss, which can be a sign of their disease, that it's really easy to monitor. They have respiratory distress. So if you're working with respiratory tract damage, it's really useful. They show lethargy and huff posture when they are getting sicker. They have the same demographic differences as human regarding age and males. They have high level of replication, which can be a good model also for transmissions. And the viral RNA can be easily and readily detected in their tract, also in their small intestine, and in both upper and lower respiratory tract. So it has almost all the things that happen to a human in a little. Also, you don't have to euthanize the mice in order to study. You can use a non-invasive qualitative and quantitative analysis in order to study the disease in the hamster. And this model is quick to grow and quick to study, and it's really cost effective. And it has been used already to study some of the famous drugs, such as hydroxychloroquine with or without azithromycin, and favipiravir, that it's an antiretroviral drug. Also in the study of some of the promising vaccines, such as this one. And to study the effect of antibodies. So you make a hamster disease, take those antibodies and transfuse them into a new hamster and check it out if it happens something and protects the new hamster. But they are not as well studied as the mice, so you may have some inconvenience as there's no that amount of literature. So that's a cons. Another type of model is to use ferrets. And ferrets are also a good type of animal model. They have a low pathogenicity and transmission levels. So they are really good models in order to study preventive treatments and prophylaxis treatments. And when you infect them in respiratory and mucosal sites with SARS-CoV. They show undetectable or mild symptoms, so you can study asymptomatic people framework of the disease using ferrets instead of hamsters. And they show some of the mild symptoms. They show it's lethargy, nasal discharge, wheezing, buildup of mucus, and sneezing. Also, they have some diarrhea. They have a fever. They don't have weight loss. They have a little of respiratory tract damage, but as they have so little symptoms, you have to euthanize them in order to study the tissue, as you cannot recognize them with non-invasive analysis. So that's sadly. With non-human primates, well, they have been studied 3 types of macaques and the African green monkey of the Bureau line that show Hector. And they show high levels of replication, but the range is really wide. And they have both upper and lower tract viral replication. They show only mild symptoms of the disease, but they can be studied with radiographic as they show some abnormalities. And they only have age demographic of the disease because the old monkeys show some shadow from the nose. That's the only sign that it's different between the younger and the older monkeys. And as I mentioned, as the range of viral replication is really wide, you can have a lot of variations in magnitude, consistency. So even though they have a high resemblance to human, they are not so good animal models to study the disease. Other type of animal models that they are studying are, they use a mink, but it is actually the same of ferret, but with a lot less information because ferret is actually a standardized animal model. Also, they have shown it that the disease can be induced in cats. They are susceptible and can transmit the disease. This is a little bit scary as they can be now some reservoirs. They have a 2% death rate. The cons of using cats is that they are really difficult to handle. They don't work with you when you're in a lab, and they are not a standardized animal model. You can also use dogs, but they are susceptible, but they show really mild degree of susceptibility and of symptoms. So it's really difficult to infect a dog, even though they are susceptible. The affinity of the virus into the host is really, really low. They use pigs, and pigs are not susceptible, but they can amplify hosts. So if your goal is to amplify virus, you can use pigs. I don't know why, but they mentioned that chickens and dogs have in silico proofs to be a host, but in real data, they have not been shown, neither in X. So you cannot choose them for vaccine production. And for fruit bats, they can show efficient replication, also as in pigs, and they show no signs except for rhinitis. So yeah, they probably have some difficulty in respiration. I really might want. And this is a table comparing all the things. And for our project, I think that the most effective model to carry out the in vivo part is to use the hamster. As you can see, hamster has the here part of the low respiratory tract infection, also in other organs. They don't show fever and nasal discharge, but they have labor breathing. They have almost all of this pneumonia pneumonia problems. They show transmission, so they can be used for infectology protocols. They show also immunology. I know that this is not part of our study, but if the resemblance between human and hamster are narrower, it's a preferred model for studying the disease. And also they, as I mentioned, they share the demographics of the human. So as they cover almost the most of the parameters, I think that it's the best model of all the things that I presented. Also, they mentioned that if you want to study in cell lines, you can use Vero, you can use this cell line, you can use the macaque, well, this cell line that also presented a Carla. You can use primary tissue or micro engineer organs or even lung organoids. And what's the importance of this study? So they mentioned that there are a large number of animal models that can use, as I mentioned, in different type of studies, and they have been established safely and efficacy, but the fact is that we need more studies in order to verify some of the animals that are not so well studied, and those animals also need new type of assays, new type of protocols. But at the end, this type of animal models will contribute to the development of all the treatment and things that we need to confront this disease. So I think that's it. Mm-hmm.
I have a question. Since I've known that there has been a lot of coronavirus zoonotic animal reservoir, one was the fruit bat, another type of bat, one was a cat, and the most important one for MERS was the dromedary camel. Why do they mention it? Do they explain why they didn't put it in? Or maybe it's because it's really hard to work with? I don't know.
About the camel?
Yep, it's still.
They don't. Even in the small studies of 10 samples, such as the cat one, it has not been shown to be neither a susceptible animal or amplifying animal for this type of SARS-CoV-2. Yep. Neither in vitro, as in chickens and ducks. In silico, sorry.
Yeah, it's kind of weird for me because you had MERS and it was a reservoir that has been already reported in literature, but I guess it's weird.
Another question?
I don't know how to make questions. Have you got any, Carla?
No, I don't have everything.
Well, in that case, I believe that we can say that we can conclude this second journal club and that we were successful in presenting the different papers, and we now have a properly developed background in order to begin developing this project for the study of biosurfactants in order to treat, reduce, or prevent the rate of infection by SARS-CoV-2, causing a viral cause of the current COVID-19. If there's anything else you would like to add, please do. If not, I think this is it for this occasion.
Well, no, guys, thank you very much. I'm going to stop recording.
Perfect.